There you go.
Great. So here we are at lecture 2, anyway, I'm continuing the lecture on CRISPR today. And remember, we described on the blackboard a model like this, which I drew up, where you have, it's a sort of a population dynamics model, where you have wild type in the clearer little ovals are wild type, where like this. And then you have spacer-enhanced bacteria. Here we're imagining a very simple scenario where you can pick up one spacer over a short period of time to make yourself immune from a particular virus. And then we imagine the dynamics where in the top two arrows are describing logistic growth, basically growth bounded by carrying capacity for both wild type and for spacer-enhanced bacteria. And then we imagine that wild type can get infected. And if they're infected, there is some chance that they clear the infection and acquire spacer, and there's some chance that instead they lyse. Spacer-enhanced ones can similarly get infected, and if so, they lyse. Okay, so they lyse, and we didn't bother putting the additional acquisition of a new spacer because these are rare events anyway. So we didn't put that in. Or you avoid the infection. That should be the normal thing that happens. Eta is a small number. Alpha, the acquisition event, is also a rare probability. And then you can have spacer loss. So that was the overall model we started with. Okay, and then we wrote this down, the same thing, as a set of differential equations. Here we are. And the differential equations, we said, describe these processes. The first line describes, the growth rates of the wild types. There's a growth process, and there are various decay processes. Either you get infected, you get killed, stuff like this. The second line tells you the same thing for the spacer enhanced. You either grow, or you get infected, you get killed, or you lose spacers and become wild type again. And then here are the processes of growth and decay for the wild type and enhanced infected ones. And then finally, you can write down, of course, an equation for the phage. And there's a parameter B that we added, which is the burst size. That is to say, when the bacteria lyse, on average, how many phage copies do they release? Question?
Yeah, the number of viruses is going to be like a whole different order of magnitude. The number of phages.
Yeah.
Is it going to be like the whole order of magnitude above the number of, because I don't really.
So the number of phages, you typically assume it can be very large to start with. It doesn't have to be. So there are many viral infections that can start with just a few viruses, but typically you assume that there's a very large number of phage floating around. At least I'll assume that, and if I'm not assuming that, if I'm assuming very few phage, I'll tell you. So in our case, we're going to assume the raw number of phage nu is pretty big, but the dynamics will change that in some way. Clearly, you can have a difference, because if the number of phage is overwhelmingly bigger than the number of bacteria, then you can overwhelm the bacteria and kill them. Basically, this is like even in human infections, the initial dose size matters. But evidence of the fact that single infection events can also matter is in the typical AIDS infection in humans, actually the whole full-blown infection eventually starts from one event, like one viral particle, right? So you can you get the initial dose, but it's only one that succeeds. Right, so even that can lead to full-blown AIDS. So these things are exponential processes. So you have to worry about these kinds of details. Great, wonderful. So I'll put that up, and it'll keep turning off once in a while. So our job to start with, yeah, question?
Why are we not considering the alpha term in the equation to the wild infected? Because we would need to have two terms that are diminishing from this growth. There will be the bacteria that are becoming resistant, that are becoming spacer.
Why the wild type doesn't have an alpha?
There, you have the wild type infected, you have the possibility of excision and possibility of lysis. Then we would need to have two terms, right?
It's because if you take the mu alpha plus the mu 1 minus alpha and add them, right, that just gives you mu. And both those transitions, one to spacer enhanced, one to lysed, gives you the same outcome in terms of the wild type population. There's less wild type.
Okay.
So if you sum those terms, the alpha goes away.
Okay.
Just by definition. Okay, very good. So our job is to solve this, as we discussed yesterday. Solving population dynamics like this is generally a complicated matter because there are too many darn equations. So typically, you can do two kinds of things. These days, you have powerful computers. So you just put the thing on a computer, use some method of differential equation solving, and you see what we get. Even if you like doing analytical things, so numerical analyses have pluses and minuses. A plus of numerical analysis is you very quickly get a picture of what's going on. You can explore many parameters. You can understand the system. A minus is that sometimes it's hard to discern an insight into what are the features of the system because you didn't work it out by hand. Some people are really expert. I used to have a postdoc who, Gaspar Tkaczyk, who's now a professor at IST Austria, who was, he could do numerical analyses and he would just vary parameters a bit and somehow get lots of insights by just looking at the figures. In fact, many biologists do, right? One of the basic techniques in biology is how to read figures for insights. So we'll try to do a bit of both, right? Except I'm not going to do numerical analysis for you here, but I'm going to draw the outcome of the numerical analyses you get. Okay, so if I now plot the number of bacteria, so n here is the sum of n0, n1, i0, and i1, It's a total number of bacteria. So I'm going to put n equals n0 plus n1 plus, I wish there was a way to tell it not to sleep, n1 plus i0 plus i1. So the kind of curve you typically see for, I'm drawing the generic curve for many, many choice of parameters, is it goes up, then it sort of comes down almost very low, then shoots up, then it sort of comes down and kind of plateaus. So we'll discuss each of these features in a minute. So if you likewise draw a picture for the viruses as a function of time, you get the following kind of features. So let me see. So this, I'm going to draw a line here, and then I'm going to kind of draw a line here. And so you start off with some number of viruses. I'm not putting units here because there's some sort of generic units. It's a generic curve I'm drawing. It'll sort of do this, and then it'll start kind of shooting up very rapidly because you see what's happening here is the bacteria are just growing. The viruses begin to infect the bacteria, right? At some point, the bacteria are pretty sick and they're starting to die. The death rate is increasing higher than the growth rate. At that point, it's really true that the viruses are taking off. So they reach this. But then the bacterial population reaches a minimum because they're becoming immune. So as they become immune, they're better able to defend themselves. So this means now the viruses can't grow on this. So they sort of drop. And then sometimes there's a little bit of ringing. And then they drop like this to about here. And then the bacterial population begins to decrease again. This is because in this model there's spacer loss. So you may have developed spacers and then you get a little bit of overshoot, but then you begin to, every once in a while somebody loses their spacers. And so the result is they're no longer immune. So now the viruses can grow on them again, but they sort of stabilize. Of course, by fiddling, if you really feel like playing, differential equations games or dynamical systems games, you can try to tune this to produce oscillations, and indeed you can. You can get oscillations here by tweaking the parameters just right, so this goes down, this goes up, then it goes down and up. You can do all kinds of things. But if you like, for some generic choice of parameters, this is the kind of thing you get. So the immediate qualitative thing that you can take away, so maybe I should talk about, this is, let me just label this. It's the initial growth, infections, CRISPR immunity takes hold, and here you have spacer loss. And very importantly, this is the thing we wanted to see if we could get without adding extra ingredients like funny molecular mechanisms and more bells and whistles. The question was, can you get phage bacteria coexistence? And indeed, essentially generically in this model, you get phage bacteria coexistence, which is nice. We did nothing except add all the basic ingredients and remembered that spacers can be lost. So previous models hadn't included the spacer loss. So this illustrates how things you leave out from your model can actually be important, because you might have thought that spacer loss happens rarely, maybe not so important. But as that's what helps to produce phage bacteria coexistence. So sometimes the details matter, sometimes they don't matter, that kind of thing. Okay, so here let me draw this. This is so this initial increase is in the phase where the infections are kind of winning. So this is where immunity builds. This is because of spacer loss. And here you have coexistence. Great. So this is, well, do I need to bother? This is actually log scale. It doesn't really matter. OK. So the fact that, so this is nice. So in other words, so we see coexistence can happen easily. So there are many things we could do here. We could analyze these differential equations, their dynamics, their ringing, their transients. But there's a whole array of tricks that you can use from differential equations theory and from population dynamics. But that's actually not the main topic here, because I know you're having other classes in population dynamics, et cetera. So I'm just going to do one thing, the easiest thing, which is I'm going to look at the steady state and try to confirm some facts about the coexistence, and then we'll move to the next topic. Okay? So let's do one thing. Let's look at the steady state.
Professor?
Yeah.
One question here. For some set of parameters, we get this kind of a qualitative behavior.
Yeah.
What other kind of behaviors can we have?
Okay. So by moving the parameters outside this sort of pretty broad range, you can get bacterial extinction, right? That's, for one example, is if you have just too many viruses to start with, or if the burst size is too big, you can get bacterial existence. Actually, I'll derive conditions for getting coexistence in a minute. Another thing you can get is viral extinction. If the immunity is really good, there's nothing to grow on, and you can essentially wipe out all the viruses. So in all of those things, actually, because we're doing a sort of deterministic differential equations kind of model, you'll actually find that extinctions won't quite occur. It's just that the numbers get very small because it's all exponentials. But so another thing I've left out of this model is this is all stochastic. So I should really be making these into stochastic differential equations, basically adding some random terms. A bacterium could die because it dies, right? Some other random cause, or or a virus could reproduce with a genetic error that wipes it out, right? That makes it infeasible. There's lots of little fluctuations going on, and I've ignored them. So really, in general, if these numbers get too small, you should expect a fluctuation to lead to extinction, right? Once you get to small numbers. So that won't actually come out here, but when the numbers are very small, we'll declare, well, I left out the stochastic part, so there's extinction. So that's another thing. You can also, if you arrange the parameters right, you can get ringing, right? So the bacteria grow, and so the viruses and the viruses are giving up, but then they lose some spacers, so the viruses run up, the burst size is sufficiently big, so that it rapidly ramps up, but then you pick up more spacers, then you get the picture. Yeah, you can get a little bit of oscillation, damped oscillations you can get. We haven't tried to look for persistent oscillations because, well, nobody's reported that, so we haven't looked for it. But these are Lotka-Volterra type equations, right? These are population dynamics, Lotka-Volterra type equations, which quite well produce all these phenomena, right? You can get limit cycles and everything. So I assume that by engineering these systems correctly, or for some system that's out there, you can see all these different kinds of phenomena. That shouldn't surprise us if that happens. Yeah? Okay, great. So, but here I'm going to look at the steady state. The equations got wiped out, so you're just going to have to believe me as to what the equations were. So if I set I0 dot equals I1 dot equals 0, remember I0 and I1 are the infected populations of the wild type and the spacer enhanced. So you get two equations. One is you find out that at steady state, G over mu V and naught. N naught is the population of, this is basically the rate at which you're getting infections from the wild type, right? This divided by mu, which is the rate at which, the infected thing changes into either spacer enhanced or or lysis. So that's that mu over there. This is just simply the growth rate divided by that rate, by the growth rate divided by decay rate, basically. So similarly, I1 is also going to be growth rate divided by decay rate. So you get G eta V N1 divided by mu. Let me repeat. Again, this is the chance that you get an infection. Well, this is the rate at which you're going to get infected because from the viruses. It has something to do with the virus's ability to clamp on to the cell surface. Eta was the, if you have a spacer already, the chance you actually get infected is lower. So this discounts for that. V is the number of viruses. N1 is the population of spacer-enhanced bacteria. So you have to multiply those things. And then you get to divide by the rate of outflow of this thing, which in this case leads to lysis. Okay? So that's easy. Great. So then if you write down the equation V dot equals 0, because the virus is stabilized also, you get an equation relating the population of spacer-enhanced cells to the number of wild-type cells. So this is interesting. This equation in N1 over N0 is the relative proportion of things that are immune in this way, in this population. And it has an interesting relationship with things like the, I have to remind myself the parameters every time because there are too many darn parameters in these kinds of things. Okay, so alpha, alpha was the acquisition rate. Alpha is the acquisition rate, eta is the failure rate, B is the burst size. So there's some relationship involving acquisition, failure, and burst size. That's kind of interesting. Some things that have to be true is that you must have beta eta less than 1, beta alpha less than beta minus 1. B eta must be less than 1 so that N1 is positive. And you also need, did I write this incorrectly? I think I wrote this incorrectly. Let's forget about this. I think I've made some mistake here. There's 1 minus alpha. So you need alpha to be less than 1. So you need basically, what this says is infection. So these are conditions that are necessary for the population fraction that's immune to be bigger than 0. I mean, that must be the case for this to make sense. So we've computed the solution, the steady state, with that assumption that there's a population, but there's just a consistency condition. So what does this consistency condition mean? It means infection of immune bacteria is rare. And this says acquisition of spacers is, of course, it's less than 1. Did I write this equation down correctly? This is wrong. That's what's going on. Woof. Okay, better. Ha. Okay, so I got to correct this. I'm sorry. This is B alpha is less than B minus 1. Now this is a non-trivial condition. So this means the acquisition is rare. Okay? So you have these two conditions, and we should set these two conditions up. Okay? So proceeding in this way, we can work out all the other equilibrium conditions. Okay, so we're going to proceed in this way. Now, the rest of this is kind of, it's pedestrian. You have to set things to 0 and solve it, but it's kind of, and it's also tedious. So I'm not going to work out the steps because it's kind of a long, page long, move this to this side, move it to that side. I'll just tell you the outcome. The important things to look at are N equals the total bacterial population winds up having the following formula. There's K. Remember, K is the carrying capacity. It was the maximum population that it could be. And then you get to multiply this by 1 minus F And F, in turn, I'm going to call this FR. FR is equal to 1 minus n over k. So FR here is the, basically kind of measures how far away you are from capacity at the end. So how many bacteria do you have? So the interesting thing is to compute this FR, and you can algebraically. And you have kappa, and I want to highlight this kappa. Kappa was the rate of spacer loss. So this is the rate of spacer loss. And we divide this by F naught. Remember, F naught was the growth rate of the wild type. B times 1 minus alpha minus 1. I'm going to write out a complicated expression to show that it can be done, and that'll simplify it instantly. There's a B minus 1 plus R minus 1, and B times 1 minus alpha minus 1 over 1 minus alpha minus eta. I remind you again, once again, this whole thing is built out of the burst size, the acquisition rate, and the failure rate. Right? Okay, so we need to derive some insight from this as to what this is supposed to mean. So I'm just going to suppose that R equals F naught over F1 is approximately equal to 1. That is to say the growth rates of the wild type and the spacer enhanced are basically the same, which appears to be more or less the case in experiments. So at least we can, to good order, figure out what's going on by setting that. And doing that, of course, here for analysis of this equation, because it makes it easier, and therefore we can sort of think more easily about it. So I'm going to write this expression down. You still have the kappa over F naught, B times 1 minus alpha minus 1 in the numerator. I'll interpret this all in a minute. There's a B minus 1 here, and there's 1 minus B eta. Okay. So a couple of key points. Coexistence requires kappa bigger than 0. Right? So namely kappa, i.e. The rate of spacer loss. That's because there's a kappa here. If there was no spacer loss, this particular solution that gives you coexistence would give you no bacteria at all. So this particular solution, this branch of the solution space would sink down to not having any bacteria at all. If there's another branch where you have no viruses at all, right? That's another branch in the system. But in this way, you would wind up with 0 bacteria unless you have spacer loss, right? So oddly, you might have thought that without spacer loss, this would be, fill up, complete. Am I saying this in the opposite way? Yes. I'm saying it in the exact opposite way. I'm sorry. F is the fraction of unfilled capacity. I'm saying it the exact opposite way. I have to confess, I got up at 3:30 to write today's lecture. So I'm not completely cogent. So okay. So let me repeat, correcting my mistake. No wonder, various people were going, okay, here we go. So the FR is the fraction of missing capacity. So that means if kappa is 0, that means you have no spacer loss. You're not missing any capacity. That means you fill up. Makes sense? The bacteria are now all immune and don't lose any spacers. Thank God the world still makes sense. Okay, great. Coexistence requires kappa bigger than 0. For kappa equals 0, you fill capacity. Fill the capacity. Okay. I should write lectures less when I'm less sleepy. Okay. So so that's one key point. So there are 2 more key points to make here, which are, actually I'll make those points later. So one thing to ask is it plausible that you have spacer loss? I'll take a question in a second. Is it possible that you have spacer loss? And indeed, spacer loss is known to happen. It typically happens at double-stranded DNA breaks and followed by, sort of homologous recombination of the CRISPR repeats, and so at a different CRISPR repeat. And so what this can do is it can lose whole chunks of the CRISPR locus at the same time. So it does happen, We haven't modeled it in a very accurate way in some ways. You know, we've talked about a rate at which you lose one spacer. You know, there's a continuous rate. So you could improve that. But all of those are directions in which you could imagine improving the structure of the model. Okay, question? Yeah?
So just on the FR equation, on the bottom term you have 1 minus B eta, B minus 1 plus R times 1 or minus 1?
This is supposed to be R minus 1.
Okay.
Which is why setting R equals 1 eliminated that term. Okay, so you know a good theory should subject itself to some test. This is a semi-good theory and subjects itself to some tests. Yeah?
Sorry, but why do we have two definitions for FR?
I hope I don't.
Well, I see FR equals 1 minus n.
Yeah. So this is a definition. FR equals 1 minus n over k. And then you calculate n, and you plug it in, and you get this.
Okay.
That's what was the intent. Yeah?
Okay, yeah, thank you.
Okay, so let's talk about some tests. So, you know, how do you test a theory? You write down some numbers. You try to figure out a prediction. So some of these parameters are measurable. So look, so if you suppose you have, so you also know that f must be less than 1, right? So f must be less than 1. So because it's a fraction. So now I can solve for eta, and you'll find an equation that you could use to test this model. It says that you can get this kind of coexistence provided the failure rate of the spacer is less than a critical value. That's just setting up the condition that FR is less than 1 and just solving, you know. It's 3 lines of algebra. That this must be true. This is equal to 1 over the burst size times 1 minus the spacer loss rate divided by the wild type growth rate, B times 1 minus alpha, alpha being the acquisition rate, minus 1 over B minus 1, plus order R minus 1. Okay? So if you look at the right-hand side, all these parameters are measurable. You can measure the spacer failure rate by, you know, testing bacteria with the spacer against viruses. The growth rate can be measured, the burst size can be measured, you know, acquisition rates can be measured. So all of this side can be measured. So you can at least check that this model was self-consistent by making sure that if you put these parameters that you can measure in the experiment together, that it is less, that the actual failure rate is in fact less than this critical value. This tells you what the failure rate, it has to be at least this low for coexistence to happen. So that's one way of constructing some kind of prediction. Okay? A second way of doing that is to observe. Oh, the second way gives you also a way of making a certain measurement. Let me tell you what that is. So the second thing you can do is you can work out that at steady state, the following relation holds. That's just dividing out the various parameters we have there. We have n1 over n0. So you work out the equation for n1 over n0 in a similar way. So this is the enhanced or spacer enhanced bacteria population divided by the wild type. That is B times 1 minus alpha minus 1 over 1 minus B eta. Again, B is the burst size, eta is the failure rate, and alpha is the acquisition rate. So one very interesting thing here is in this steady state, the spacer loss rate doesn't even appear. Right? It just depends upon the other parameters. Now that's also something on the right-hand side, everything can be measured at steady state. And on the left-hand side, everything can be measured at steady state. Right? You'd have to check the spacer enhanced versus just wild type. You can't just do optical density measurements. You'd need to put some kind of marker, like a fluorescent marker for the things that have the spacers there. You know, you'd have to do something like that. But if you do that, then you can measure this. So you can check whether or not this theory is correct. And surely you'll find that it's not quite correct, right? Because I've left out things. But what you're hoping that it comes close, right? The art of doing this kind of modeling is you want this kind of model, which is, you know, we just went in there and just put in all the basic stuff, right? And the basic stuff should get you into the ballpark. No chance it's going to be exactly right. And then you adjust it in whichever way you want to capture whichever phenomena you care about in greater detail. Okay, all right? So let me, yeah. You have to kind of,
Sorry, to go through this equation again, please.
This one?
Yes.
So I didn't actually convey any particular insight about this equation. I think that's what you're asking. There's some complicated set of formulae here, right? So what is the point of it? And the point of, so I didn't actually describe any why it had took this form. We can analyze if you like, right? So basically, surely the bigger the burst size, in order to get coexistence, the lower your failure probability should be, right? Because if you have huge bursts, your spacer needs to be really good because you're making many, many things at the same time. So that explains that. And we can go through each term like that. It's not the most important thing for me, so I'd like to just continue because I'm realizing that there's quite a lot I want to cover, if that's okay. But we can afterwards, during question time, if you want to talk more, I'm happy to. Okay? So the next thing I want to talk about is the diversity of the spacer repertoire, right? So, this sequence of lectures is all about thinking about adaptive immunity and in particular about CRISPR, and I'm going to be interested in the structure and organization of the repertoire of immune elements, right? So far we've been using a dynamical systems approach to get some insights into that, what a steady state, can you get bacteria, etc. But now I'd like to sort of use this dynamical model to dig in a little bit more into the diversity of the repertoire and how the dynamics may control whether you have a diverse repertoire or a narrow repertoire. Part of the reason I care is this is something that in experiments you care about. You do experiments and you find, well, you do an experiment in a lab and you find that the repertoire of spacers the bacteria pick up is kind of skewed and pretty tilted towards some spacers. And then if you look at wild-type bacteria, they tend to be more spread out and less peaky in some direction. So we'd like to understand where this is coming from and what these differences are, what they mean. Are there knobs the bacterium can tune to go between sort of peaky immune repertoires versus flat repertoires? So let's try to investigate. So the first thing we need to do to discuss that is we need to discuss a model with many spacers. So, in models with many spacers, many interesting phenomena can happen. So, for example, there's a thing called priming, where you can go and meet a virus, and your spacer doesn't quite match it, misses quite a bit, but it's kind of approximately right. And in some CRISPR systems, then what the system will do is it'll latch on and then snip a new spacer right next to the partial mismatch. So, and that can, produce effects, right, for example. So that happens when you have multiple spacers that you're gathering. So there are many effects of this kind. I'm going to ignore them all and make the minimal extension of this model and see how far I can get, and then we can keep adding more bells and whistles. Okay? So unfortunately, this is gone here. So I'm just going to rewrite the equations. So there were 4 equations. What's that? Yeah, I'm not going to worry about it. I don't know how to do it. So, do you want me to write the equations out again, or just, I'll just write them out. It'll be quick. So I'm going to rewrite all these equations. There's F naught. So I'm going to make F. So I'm going to make some changes with what we had before. Remember, previously we allowed for the possibility of different growth rates for the spacer enhanced and the other one. Well, now if I did that, I need to allow for different growth rates for the different spacer enhanced ones. In any case, these things are about one, they're about all equal. So I'm going to make them all the same, right? So I'm trying to increase some complexity and therefore decrease something else so they can still handle the equations, right? Again, for the philosophy of this kind of modeling is I'm not trying to get exact model. I'm trying to get a model that captures the qualitative effects so that I can understand them in some way. So now, it can be that the number of wild type increases because of spacer loss in any one of the different spacer enhanced bacterial types. I'm going to let I equals 1 through n for the different kinds of spacers. So then I can put a sum I equals 1 through n, right? And again, I took this to be that all of them lose spacers at the same rate. That's also not completely obvious. You know, there could be many, many differences. And I'm pointing out all the places where the model could be modified if things don't actually capture what's going on, for all the places that are kind of loose in what we're setting up. So this is coming from the addition to wild type by spacer loss. And then you have G, V, and naught. This was the same infection rate. So now I can talk about enhanced bacteria. And they will have a similar equation. I have Ni dot is equal to F. I'm using the same growth rate, 1 minus N over K. I'll put an Ni here because now each spacer enhanced thing, you know, grows separately. It's a kind of a lineage, if you like, that of the bacteria. I get to subtract the effects of spacer loss. This time I don't need to sum over I because I'm just talking about this species or this lineage, right? Now, for example, I could agree that there are different failure rates for the different spacers. The reason I care is that's going to be one of the phenomena that could make a difference between which spacers you keep, right? If one spacer is good for eliminating virus, you probably want more of that, or you probably prefer that to one that's less good, right? So we should keep that to be different between the different spacers because we're trying to study that. Okay, and then we have plus alpha I, mu I naught. And I'm going to set up alpha equals sum over I of alpha I. So alpha, remember, is the acquisition rate. And I'm putting this here because another thing that people wonder about and have studied is maybe some spacers are easier to grab. Because, you know, in the end, you have a molecular machine. Right? It goes like this. It sits there. It goes snip, snip. You know, sometimes some bits of DNA will fit better in your machine than others. So that might make some spacers easier to acquire. Or it may be that if I look at the entire viral DNA, a particular sequence appears multiple times. So if that happens, you're kind of more likely to get it. So it might well be that some spacers, some sequences are, you know, more frequent or more easy to acquire than others. So we should keep that as a parameter there too. So we put in different failure rates and we put in different acquisition rates. So we're going to study the balance of what different failure rates and different acquisition rates may do to the diversity of the immune repertoire. Okay, and then of course we have the I1 dot, I0 dot, that is wild infected, and this is equal to, in the same way as before, G, V, and naught. That's the infection rate minus mu I naught. This is the rate at which they transition out either into having a spacer or dying. And then there is the enhanced infected. This is again the same equation that we had before. And the equation is going to be I1 dot is equal to, and then I'm going to have an eta I, because that is the failure rate, G, V, and I. So this is the rate at which, you know, the enhanced species I gets infected, right? So I put these things in. And then I'm going to put a minus mu II, which is the rate of failure, of death of this thing. Okay, and then we can put in the virus. And let me put the virus, V dot is equal to B. And now I have, you know, this arises because the wild type die. And I have a 1 minus sum on I equals 1 to N alpha I. So what's going on here is that either the virus, you know, it gets infected, and then the virus either dies producing bursts of size B, or some fraction of the time it acquires a spacer, right? So this is the total spacer acquisition rate, the parameter alpha that we wrote up there, and that sort of reduces, you know, the increase in virus after infection. So that's what that's about. And then you get a plus B mu sum on I equals 1 through NII minus GV N0 plus sum on I equals 1 through N of Ni. recall that this also arises from the death of the immune viruses because they actually got infected, and this arises from viruses entering the cells and infecting them, right? So you lose viruses. Great, so that's our setup, and we need to work out the same kinds of things. So I'm going to be interested in coexistence again. We already talked about many other phenomena that could happen, but I'll just talk about coexistence. And we can solve for coexistence in exactly the same way. That is to say, we can set all the time derivatives to 0, and the manipulations are clearly going to be very, very similar. There are some now sums over all the different species of the bacteria with different spacers in them, but other than that, it's very similar. So coexistence, and I'm going to say solve similarly because I don't want to waste your time with repeating, the same kind of algebra again. You can perfectly be able to do that yourself. And if you have F equals 1 minus n over k, the fraction of unused capacity, this turns out to be, as before, kappa over F. F is the growth rate, kappa is the loss rate, remember, of spacers, same kind of thing. And then you have a B, 1 minus alpha minus 1. Alpha, remember, is the composite acquisition rate divided by 1 minus beta. Now there's a slightly different thing, eta bar, B minus 1. Let me tell you what eta bar is. So eta bar is equal to the average failure rate probability. And that is equal to the sum on all the species of Ni, eta I, divided by the sum on all the species of Ni. Okay. So in other words, it's exactly like the previous expression, thank goodness. It was sort of minor change, you put some averages and some sums in some places. Excellent. So we are interested in the predicted diversity of the immune repertoire in this model. Right? That's basically so that we can get some handle as theorists on the parameters that may control how diverse the repertoire is. Part of the role of theory is to give experimentalists targets for things, or likely targets for things to study. That's because experiments are very hard, very expensive, and take a hell of a lot of time. Right? And therefore, it's a good idea to have some sense before you start of which things are likely to give you results. And that's where one of the ways in which theory helps. It helps come up with hypotheses for the knobs you should turn or look at. So let's see if we can use this model to think about that. Okay? So we're going to look some, we want to look at the predicted diversity. So you have to do some algebra here. Again, it's pedestrian but tedious, so I'm not going to do it because we would spend half an hour just pushing symbols around. And what you find is three things. So first of all, I'm going to write down three equations. Do I need to write this equation? Probably not, but too late, I wrote it. So here's equation 2. This equation I do need. So I'm going to look at Ni over N naught. Okay? This is the important parameter for us because it tells us, compared to wild type, how many do you have of the different spacer-enhanced species? So that's really, so if this is completely even, if these things are all equal, that means you've got a uniform population. It's a very even and diverse distribution. If this thing is very sharply peaked, it's not a diverse distribution. So this is the object we really want to study. So this is equal to alpha I B F. So F, remember, is the growth rate. B is the burst size. F is this thing, right, that tells us about the fraction of unused capacity. I get to divide this by kappa, which is the failure rate, minus F, growth rate, fraction of unused capacity, 1 minus eta I, which is the failure rate of this thing, so on. So this is a slightly complicated expression, so you can imagine the amount of algebra to find this out is correspondingly, it's like 2 paragraphs instead of 2 lines. So there's a little bit of work to do there. Anyhow, so we know what all these parameters are, and just for kicks, I'm going to write down a third one, which is if I take a sum on I equals 1 to n of Ni over N naught. This is the analog of the object we looked at before. How many bacteria are spacer enhanced relative to wild type in total? This has a slightly easier expression, as it turns out. It's B times 1 minus alpha minus 1 over 1 minus B eta bar, which is very similar to what we had before. So one thing about this thing is the reason I wrote this equation down is that this is a way of measuring the average failure rate. So, you know, if you're doing an experiment with 30 spacers, it can be expensive and hard to measure the failure rate for all of the individual species separately. But if you can just measure by sort of lighting up the different kinds of bacteria, right? Because to measure failure rate, you've got to track a bacterium with a particular spacer that you know you've tagged it, you know which spacer it is. And then you need to say, oh, look, it failed, and how many times it failed. You can imagine this is complicated. You could do this for 1, but you have to do this for 30. This is, you know, graduate students have lifetimes and need to eat dinner, you know, things like this. So keeping that in mind, here's the measurement you could make. You figure out by lighting up the different bacteria, how many of them are immune enhanced, so how many of them aren't. You take this ratio. You can measure burst sizes easily, acquisition rates, right? The total acquisition rate you could try to measure, and then and then you could read out from this. You could measure the eta bar, right? So in this way, you can get a handle on this average failure rate, which is an interesting parameter. You know, how useful is immunity is what that characterizes. Okay, now I want to look at this equation 2 because that gives you actually 2 predictions that are of interest to us concerning 2 qualitative predictions that are of interest to us. So the first statement is that the steady state population is proportional to alpha I. So what that means is that if all else is fixed, it's very difficult to get large differences in the populations of the different spacer types, or the different, having different kinds of spacers, just from changing the acquisition rate. If there's a 10-fold difference, you need a 10-fold difference in acquisitions because it's proportional. So all else being fixed, the acquisition rates are going to tend to not give you a particularly peaky population. So this means if all else is fixed, a peaky distribution of spacers needs large differences in acquisition rate. So this is one of the standing hypotheses for how you get diversity, that you have differences in acquisition rate. And this shows you have to work very hard if you need to get very sharply peaked populations this way. But maybe it's useful. But on the other hand, if you sort of, okay, I'll say more about that in a second. So B, notice that eta I appears in the denominator. So remember, eta I is the failure probability. So this means small differences in efficacy can have large effects. That's because of the denominator. Notice this thing appears here, right? So if you have this thing, this is balanced in some way. By changing eta I by a relatively small amount, you could change the denominator. And if you change the denominator, you can have a bigger effect on the outcome. So this suggests that when you see sharply peaked populations, the sharply peaked immune populations are being basically governed by differences in efficacy of these things. And in a lab experiment, you take you take a bacterium that's naive, you've emptied out its CRISPR cassette, and then you sort of probe it against the same virus again and again, right? And in that condition, you could imagine that the thing you really want to do is to get the best spacers, because it's the same virus that's coming at you again and again. And so you could imagine that in a sequence of serial experiments, that's what you see. You wind up seeing that the best spacers that give you the best defense dominate the thing, and you get this very peaky population. But you could likewise imagine that if you're in the wild and you're not getting tested again and again in this way, that what actually happens is that the differences in efficacy between different spaces don't matter so much because, it's one and out, right? A virus comes in, your population fights against it for a bit, you pick up some spacers, you're done, then you go on to the next one. So it's really the acquisition rates then that matter, right? Another thing you can show here, which I haven't quite shown, is that if you tweak up the overall alpha, if you tweak up the scale of alpha, and you just tweak up the acquisition rates, it tends to push you towards a flatter distribution. That's because all the alphas go up, right? So all of these things are going up. But there's a bound on how high they can go because they have to sum up to n naught, right? So if they're all going up, they'll wind up evening out in some way. So actually, but just so you could imagine that one way for bacteria to tune between more highly specific and more diverse repertoires is simply to tweak up their acquisition rates. And there are mechanisms for doing that. For example, you can upregulate the production of the Caste proteins that are responsible for getting spacers to you. So you can tweak up your acquisition rate, and it's known that during infections, bacteria do that. So there's ways in which you could imagine looking for that bacteria tweak the level of diversity that they have, or it's even possible that the acquisition rate itself is under long-term selection, right? That there's some balance between picking good spacers and picking a diverse repertoire over, over ecological times, and that gets regulated or selected for over time. So that's an interesting question you could ask. So that's the kind of question I'd like to ask. So typical acquisition rates lie in a certain range. They have it's not insanely high, it's not insanely low. In fact, it's not that high. So what sets the operating point? Well, you could look at the set of balance between diversity and specificity and try to see if that diversity-specificity balance somehow leads to the particular level of diversity that you that diversity-specificity balance for defending broadly and specifically, maybe that leads to the particular acquisition rates that you see. So I'll put that up also here, and that's actually the last point I wanted to make. So there's a trade-off here, which is one of the things that we would like to think more about. So you could have small, you could have a skewed repertoire. And this is useful for strong, specific responses. And you could have a diverse repertoire. And this is good for broad defense against the virome, which I mean the sort of total viral population. And so the question is, how should you balance these considerations? And also, is the balance tunable? As the bacteria change circumstances, right? During the lifetimes of a colony, as the colony progresses, are there knobs for it to be able to tweak between specificity versus this, depending upon how things are going? And that would be very cool, and it's probably there. It would be a good thing to have. Okay, so that's that for this particular kind of modeling. Okay, so the next thing I want to do is I want to say, great, we have some sense here of how the dynamics of the immune system works, of this immune system, of the CRISPR immune system works. Our model didn't contain, all the features of the system, left out many things, the different CRISPR types, details of exactly how the binding works, no priming. I could go run through a list, but we've already got some reasonable insight into how, some of the main parameters. So next, I want to push to the next level of question, which is that one of the things that we didn't talk much about is how much memory should you keep? Okay, so the point I'm going after here is we just said, oh yeah, and you pick up spacers, you do this, you do that, and the dynamics gives you a certain amount. And then you have some loss rate. So because you have some loss rate, probably you're growing your CRISPR cassette, and then you have some loss, so probably there's some balance there, and it settles down. So actually, there's a puzzle associated with this. You can think about the number of spacers the bacteria keep as the depth of immune memory. How much memory should you keep? And whether you're talking about animals at the large scale like us, or in this system, the amount of memory you keep in a system is actually a very interesting and problematic question. That's because you would naturally think that having lots of memory is good, right? Because then you can use it. But then you need the machinery to use lots of memory. So if you don't have a machinery to use lots of memory, and you only can look at little bits of memory at a time, having lots of memory poses an opportunity cost. Because, well, you can't look at all of it. So you may look at the wrong piece of it. So there's clearly some sort of trade-off. And even in things like machine learning and human learning, people talk about the value of forgetting. And so, okay, so there's a bunch of stuff I'm going to say about that, but there was a question there that I didn't take.
Okay, the question was just before we moved on. Could you clarify what's a peaky population and a flatter population?
What I mean by that?
Yeah.
Okay, so suppose I have, this is the Ni over N, Ni over N naught. This is I, and this is I here. So these are all the spacers. What I mean by peaky is this is peaky, and this is flat. Okay? So they're all even. Now, this is misleading because what I've done here is I've ordered it so that it looks like a peak. Really, there's no ordering to the spacers. So what we mean, it's very jaggedy. And this, if I draw it in some random order, it's very jaggedy. But we can always order it by biggest to smallest, and then you'll get this. Okay? Okay. So now I'm going to start out, so I'm going to sort of illustrate, at least for myself, the way I come up with questions to actually think about, right? So it's clear that there's some weird thing going on about how much memory you should keep, and sort of in very general grounds, there's some issue there. So the next thing that's useful to do is to actually look at, what does the data say about how much memory the bacteria keep. So in lab experiments, you find CRISPR repertoires are small, and you'll find like 1 to 10 spacers, not very many. So very, very little memory, okay? And maybe that's not so surprising because, you clear out the CRISPR locus, you stick in, you probe it against a bunch of viruses. How many of these things do you need anyway? There are plenty of studies that show that if you just have a couple of spacers against a virus, you're pretty good already. Of course, you're going to get escapers. The virus is going to escape, so you need to pick up one more. We'll say more about that later. So maybe that's why. Not clear, right? But maybe that's why. So wild type have more. Let's say they have like dozens to a few hundred. Depends upon what species you look at. Okay? So one thing you might say is, well, maybe they have hundreds of spacers, but actually they don't use them, right? Just because they have them doesn't mean they're used. So let's ask the question, are they used? Okay, and you can check this through RNA-seq measurements and things like this. Okay, so there are many facts that come to light. One is that there is some decrease in expression away from the leader end. So it's true that to some degree, the more recent spacers are more, expressed and more used, therefore, to construct defenses. So it's true that some of the older spacers are kind of a little bit less used. So B, the decline is graded, so it isn't necessarily that sharp. But there can be internal promoters. So I'll explain what that means. So remember that in gene expression, you can need a promoter to help expression of the adjacent gene. So sometimes CRISPR loci have internal promoters, so the expression level sort of decays a little bit, and then boom, goes back up again because there's an internal promoter that helps the expression machinery to latch on again. So in fact, in that case, it's not true that when you go deeper in, it's declined because it's back up again in this way. This is all just to justify that we should take seriously these hundreds of spacers, few hundred spacers as a few tens to a few hundreds of spacers as being the operating point of the system. That's known. And so this increases expression of sort of distal spacers, distal far away from the leading end. And you can even have reverse sense transcription. So that the CRISPR locus is transcribed on both sides, which means that, again, it would be misleading to say that end is less expressed because it's not the leader, because, both ends are the leaders in some way. So I'm going to take all of this to mean that, if you look at wild-type bacteria actually interact in the environment, we should take seriously the idea that, there are a few dozens to a few hundred, at most, spacers or immune elements in the immune system. Okay? So this raises to mind a puzzle. I'm going to write the puzzle down. And the puzzle is, why do bacteria maintain such small immune memories? Memories given two things, given A, over generations, they have met thousands of species of phage, at least. And B, there's plenty of room the genome has, like millions of nucleotides. So it's not like there's any shortage of space. You could stuff a lot of memory in there, and you choose to remember like 100 viruses. That's nuts, right? If you own an immune system, but you don't use it, right? So I'm going to try to, I've already presaged the answer that I'm going to try to give as to why the immune system is not as big. But still, it's a puzzle if you look at it first as to why you have so little why you maintain so little memory in this immune system. Let me contrast that with vertebrates. You have millions upon millions of immune receptors kind of floating around in your body. Yeah, there was a question somewhere.
Just a trivial thing. When you were splitting into many users there, the second one, decline in graded, the B.
It should be decline is graded.
Oh, okay.
What I meant to say by this is that decline is gradual. I mean, it isn't like, you go past the second spacer and there's no expression. It's sort of like this,
Okay, perfect. Thank you.
Yeah. Okay. So, when you're faced with a question like this, so this is the kind of question I actually personally really like, because there's an actual puzzle about the organization of a system. So usually, you remember when I was talking about physics of living systems, one of the things that's different about living systems from regular matter is that living systems that sort of have a purpose, right? They do stuff for a quote-unquote reason. There's some behavioral output that's required. So I'm going to ask first the question, what is CRISPR immunity for? And we will pose two hypotheses which will lead to different answers for what you think, how big you think the repertoire should be. So here's hypothesis one, which is very reasonable. And this says that CRISPR exists to defend against phage in a dynamical co-evolutionary extended encounter. So the idea here is, you're a bacterial colony, in comes a phage. The dynamics that I was describing earlier happens, right? And you go down, you go up, you start doing things. But then the phage are co-evolving with you, so they escape. Now you can get infected again, so you got to do something about it. So the CRISPR machinery does its thing. But of course, there are some of the old phages still sitting around. Not everybody's mutated, so you need to keep that around too. So you grab another spacer. Now you've got two spacers. You're trying to defend against both, but another escaper comes. So it keeps going. It's like COVID, right? You were on COVID, gamma 0.12, 0.8, or whatever it is now. Whatever. So that kind of thing. So it's for that. So that's one very reasonable hypothesis, that you have this because you're engaged in this sort of combat for a week, 2 weeks, and you have to keep going and going and going and going until eventually you beat the thing down and its ability to escape your immune system declines, or it becomes less virulent. And if the burst size for whatever reason drops to like 2 because this mutated virus can infect you but can't actually produce many, can't produce much output, well, maybe that's okay, or maybe it can't infect you as easily, so therefore it's okay too. It's less infectious. Maybe the current stage of COVID, for example, is like that, right? It's actually infectious, but I guess the current stage of COVID is more infectious, but causes less harm, as in, nobody's dying. Well, people are dying, but you don't die as easily as you did at the beginning. Okay, so that's a reasonable hypothesis, and let's see where it takes us with thinking about this this problem. So several authors. Oh, I should list some papers, I says. I didn't list papers for this. So for this work, the relevant, this is mostly taken, I learned these ideas from a paper that I wrote with Serena Brade, Maria Busaglia, Tiberiu Tessilianu, and me. And this appeared in PLOS, QBio in 2017. You can see more details there. This work is in a work by Serena Brade, Armita Nur Muhammad, Siddhartha Goyal, and me in PNAS in 2020. Okay? So you can see more details there. Now, several authors, and there are too many to list here because there are many people have done this, have studied the population dynamics of this. So many people model these scenarios, and the models are akin to the model that I described earlier. Remember, I set up a population dynamics. Now, these authors have that kind of population dynamics plus coevolution. They imagine, for example, they make some little model of what the viral, what a particular strain of the virus is. Then they imagine that it mutates and becomes another virus. Then you pick another, And so there's a method of sort of modeling this kind of thing. And so they use that to study the dynamics of the system. So definitely more complicated than what we did. But goes towards the coevolution thing that I was talking about. Okay? And they found 3 things. So in this kind of scenario that you have this coevolutionary chase going on between bacteria and viruses, recently acquired spacers are most useful. So indeed, in this case, the things in the leader end of your CRISPR cassette are the most useful. That's because the virus is mutating. So very quickly, if there's a better strain for some reason, that's the one that dominates. So, well, you know, so you really want something to defend. You want immunity against that better strain. So that's definitely true, and you can work out the population dynamics of that. B, interestingly, CRISPR is not useful at all, actually. It's a waste of time if the virus evolves too quickly. So there's something intrinsic about the mechanism of CRISPR. You know, the timescales involved in the binding and clipping and incorporation and expressing Caste and, you know, all this kind of stuff. Because of all of those timescales, if the virus is really rapidly evolving, CRISPR does almost nothing to defend. You really have to rely on innate immunity. And as a result, if you also hypothesize, as sometimes people do, that having CRISPR imposes some kind of cost on the system, it's not clear if it does, but if it does impose a cost on the system, then for very fast evolving viruses, you would actually select CRISPR out. And there are ways of doing that. You can either remove, for example, there are anti-CRISPRs. There are ways of disabling the CRISPR-Cas mechanisms. You know, there are many kinds of things you can do to turn off CRISPR, which I will tell you a little bit more about later. And the third thing you find out is that suppose you're not talking about a virus that evolves too quickly, then you find out that prokaryotes need just 5 to 7 spacers for an optimal defense. Basically, having more than this doesn't help you. If anything, it wastes your time. And it wastes your time if you have more than this, because sometimes you wind up expressing spacers, you know, using Cas proteins to express spacers that are not useful anymore. So having more than 5 to 7 is not so useful. Having less is not so great, because you need to deal with enough kinds of viruses. And so 5 to 7 is about enough, which is consistent with the lab experiment scenario where you don't really have that many spacers. But certainly, this kind of population model clearly does not explain the size of the actual repertoire of the wild type cells. So I'm going to put up a second puzzle. So the first puzzle that I wrote up over there was, why is the immune repertoire of bacteria so small compared to what you would think it should be? But now I can write down a puzzle 2 in the context of this kind of natural set of models that, or this natural hypothesis that CRISPR is for this, to deal with the coevolution of viruses. And write down, if hypothesis 1 is true, why are natural, by which I mean sort of out there in the wild, CRISPR repertoires so big? So we have 2 related puzzles here. On the one hand, it seems like it's an insanely small amount of memory compared to the number of viruses you probably have to deal with, and compared to how much space you have. On the other hand, it seems like it's sort of crazy big, like 1 or 2 orders of magnitude bigger than what you would need if you were really, every time a virus comes in, you know, you chase after it and you get rid of it. If that's what you're doing with CRISPR, you just have too many of these things. You know, so what's going on? So here's a clue, again from the data. It turns out that older spacers, sort of far away from the leader end, often correspond to evolutionarily conserved regions of persistent viruses. So that certainly sounds like a useful thing to have. Viruses, because of the way they function, some bits of their genomes have to be pretty conserved because they use them in some very important way for some function. And so, of course, that's a good spacer to have because you can nail the old ones, nail them that way. And so the old spacers, in fact appear to be useful in that way. Right? They're keeping a memory of the stuff that's really good, really useful to have. So this leads us to an alternative hypothesis that we are going to study. And hypothesis 2 says that CRISPR is primarily, it could do that too, it could do the coevolution too, but I'm going to suppose that it's primarily a long-term memory of the virome and so the idea is basically its goal, what you really use CRISPR for, is to protect against reinfection by the original strains that used to come and get you. But now, they've survived somewhere in some other niche, and then they come back, and you've got to still protect against them. Even if they've mutated a little bit, well, you still have something that kind of matches them. So because it kind of matches them, you can, you can do priming and sort of grab a nearby spacer. So it'll help you at least to do that, and then you're set. You can deal with the new virus. Right? So that's the idea. So in other words, CRISPR is, I'm going to say this again, is long-term, not short-term memory. In this sequence of lectures, we don't have time to talk about this, but if I was going to lecture about memory in sort of vertebrates or in humans or something like that, we also, for example, have long and short-term memory. We have long-term memory, that's forever. Short-term memory, that's for a day or two or until the next exam. And then we even have working memory, right? You can remember about 7 things, and then 30 seconds later you can't remember it. So we use different kinds of memory for different kinds of purposes. So it's entirely possible that even in this system, if you think about it as a memory, that general rules of how memory works can matter, right? Okay. So we do, however, have a constraint when we think about this. And this is the constraint I mentioned several times already, that for memory to be useful, you have to be able to employ it. In this case, in the defense machinery. So in other words, the whole Caste, transcription and Caste machinery that hunts for viruses needs to be able to, read this memory and use it. And you can imagine if you've got, so many things that can read the memory, and you've got, say, way too much of it, which piece of it are you going to read? Right? You have an enormous amount of memory, and I'm a Caste protein. I need to pick up which of these things to use. I don't know. I'll pick one randomly. And you can imagine this is going to produce an effect. In fact, we can almost, before we even do anything, see that there's going to be a trade-off. If you have too little memory, you're going to have that. Actually, let me write it out. So we could say that there should be a trade-off. And we are going to study this trade-off. And the trade-off is between deep memory, which gives you wide coverage, and versus shallow memory, which should allow you to mount an effective defense. Okay? So we're going to evaluate this trade-off. So one of the things I want to do is, in this sequence of lectures, is kind of illustrate different styles of modeling, right? Because there's many ways of solving problems. That's part of the thing I promised at the beginning. And one of the reasons I chose the CRISPR system is because in this sequence that I'm going to work through, we're going to use a very different style of there's going to be no population dynamics. We're going to try to use probabilistic reasoning and optimization to try to figure out how we think it should be, the short-term memory should be organized. And then I'll do a third unit on CRISPR. I've sort of slightly changed the plan of what I'm going to cover. And tomorrow, which I'll be covering that partly tomorrow, in which we'll blend these two kinds of ways of modeling things, both statistical modeling as well as a little bit of sort of dynamical systems, and use that to study another interesting phenomenon in CRISPR. So here's our model. And the model is CRISPR as a probabilistic long-term memory. So it would be good if you took this down, because tomorrow I'm going to need this again. And so you should keep all the parameters, I think, in place. Well, actually, no, you know what? You should take notes anyway, but I'll put it up again. So the first assumption, or these are not assumptions. I'm sort of writing my parameterization of the problem, right? So if you're a theorist, you need to think about what are the factors that matter in the problem. What are the things that matter? You need to give them all names and parameterize the thing. Then you need to write down the relationships between the objects, and then you calculate, right? That's our standard, form of doing a theoretical analysis. So we're going to agree that bacteria encounter J equals 1 through K types of phage. With probabilities Fj. So I should give you full disclosure. I'm going to set this up as a very general problem and write out a very general expression. And then almost immediately, I'm going to make all kinds of approximations and simplifications, and I'm going to say you encounter all the phage equally likely, etc. That's partly because there is an open problem, which is solving this for the actual systems within a skewed distributions of phage, etc. So I want to set up that problem, even though that's not what I'm going to solve. Partly, we sort of immediately specialize this as sort of with lots of assumptions, because many of the parameters, as you'll see, are not known. Right? So one of the goals of theory is to set up frameworks for thinking about systems which guide what should you measure, right? What measurements would help you understand the system? So you'll see there are various kinds of measurements that would be good to have for building this model, and maybe those are things that one could encourage experimentalists to think about. So bacteria encounter 1 to K phages, and these with probabilities Fj. By the way, that's not data that's really available. Who has gone into the wild, into some pond somewhere, and sampled how often you've encountered different kinds of viruses? There are a few studies like that. So for example, Devaki Bahaya, who's a professor somewhere in a research institute near Stanford, goes to the hot springs at Yellowstone, and she samples them, and then sequences all the bacteria and the viruses there, and she does it periodically, every once in a while. So we have some kinds of longitudinal information like that. But you could do other things, right? If this was really important, if you wanted to sample the virome, you could take an environment like the hot springs or whatever else you want to do, and just keep a probe there, like a little robot that every day takes a little sample, does the sequencing, another sample, sequencing. It's a question of what questions are most useful to study given the limited resources that we have to do. Anyway, but we should keep our minds open, because it may turn out these are really important questions to study, and therefore we should do it. Okay, cool. Anyhow, so the next thing I'm going to suppose is I'm going to say that the CRISPR loci have L spacers. So that's just a parameter. And I said CRISPR loci because although in the previous section I mostly talked as though there's one CRISPR locus, it didn't really matter. In fact, many bacteria have more than one CRISPR locus. So this is, in total, how many spacers do you have? The third parameter I need is I'm going to let NP equal the number of Cas proteins. This is going to be a very important parameter for us. So every cell is a little machine, and every machine, it has lots of little components moving around, and these in a cell, these components come with various copy numbers. It's no good within a cell to have one copy of a given protein, because you don't know if it's going to make it to the right place at the right time. So you need a certain number of them. Right? So the typical number in a cell, the size of an E. coli or a bacterium, is going to be like 1,000, like a few thousand. So ballpark, we're talking NP is a few thousand. Right? I'm going to take it to be of order 1,000. Okay. So then, but we're going to keep these parameters free, because of course, although I'm going to try to make generic ballpark predictions to land somewhere, the way you really test a theory, as we will discuss later, is to understand the variations around the mean prediction. So E. coli and Vibrio are different. Right? They've got different details in them. So maybe if you could, a real test of a really great explanation for this kind of thing would be to see how the different, how things differ between the different bacteria. Okay. But anyway, we won't do that. So next, I'm going to suppose that you have a spacer configuration And as you'll see, I won't use much of this technology at all. But if you wanted to set up a theory of all of this, what would you do? You'd need to think about the spacers. And you might say the spacer configuration is a vector. I'll call it S1 through Sk, where Si is the number of spacers of each type. Right? So these are supposed to be spacers that are specific to each of these different kinds of viruses. So I just count how many of these you have, and it is what it is. The next thing you could imagine is, well, it's possible, and we're going to simplify this almost instantly, it's possible that when you're infected by 2 viruses at the same time, right? So, well, we could then model that by saying the phage configuration in an infection that you might have is V equals V1 to Vk. Vi equals 1 means present. That phage is present. And this is 0 is absent. I'm basically going to argue that infections are rare and sparse, and therefore you basically can take this to be a single one somewhere in this, right? So I didn't actually need this formalism, but I'm setting it up to show you that we could write down this problem in general and attempt to solve it, right? At least numerically, we could attempt to solve this problem more in general. And then finally, I'm going to talk about, this is also going to be important for us, the configuration of Caste complexes. What do I mean by this? The key point for us is going to be just because you have so many spacers of type 1, type 2, type 3, etc., doesn't mean that when you've expressed them that you have so many Caste complexes of each type, right? Because it is a statistical process as to which spacer gets attached to which Caste. So we're going to work out that statistics, and then we're going to talk about the Caste configuration. You know, how many Caste complexes acquire a particular spacer. Actually, some may, yeah, right? So that's the kind of thing we're going to go after. So the configuration of Caste complexes is some vector, D, which is D1 through Dk. And that tells you, and Di is equal to the number of complexes specific to each phage. Okay? So now this is the total thing, right? With this information, I should be able to write down a complete model. I have 3 minutes, so I'm going to use them. Because I can, in fact, write down the model in 1 line. And then we'll discuss this at the beginning of the next lecture. And then we'll simplify it so that it's something I can actually do on the blackboard as opposed to being able to, you're going to eat up my 3 minutes.
This phage configuration and Caste configuration.
Yeah.
Each bacteria has one?
What's that?
Each bacteria has one configuration?
Right.
Or
So we're talking about, the way I'm going to think about this is I'm going to think about an individual bacterium. I'm going to ask what's the probability it survives. And so I'm going to write down the probability. So there's going to be some distribution over D and alpha. You know, I will write this down in general. And so each bacterium, so there's some chance. So given what spacers you have, there's some chance that you have for this, right? And so I'm going to argue that this problem can be solved in general. But to do this on the blackboard and do this easily on the blackboard, I'm going to specialize in various ways so that we don't need this full power. But let me write down the general model first, and you'll see. Okay? So this is our model. It's not even a model. I just want to write down the probability of survival, right? The idea is that the CRISPR organization should be selected over evolutionary time to help you maximize your probability of survival, right? That seems like a reasonable idea. So then I'm going to write down an expression for the probability of survival. Somehow I found it easier to write down 1 minus the probability of death. So I'll write that. After having written it down, I think I later realized that I could have just written down the probability of survival directly. But anyway, I'm going to write it down like this. So the probability of survival is 1 minus, well, let's see. I'm going to sum over all possible viruses, viral attacks that could come after me, PV of V. This is going to be the probability that you get this particular viral configuration attacking you. Then I'm going to sum over all possible spacer configurations, right, such that the total number of spacers is L. So I'm going to have a certain number of spacers for virus 1, a certain number for virus 2, etc. And I'm going to write down a probability of the spacers. Here's the spacer vector, the configuration of the spacers given the length. Okay? I'm going to multiply those things together. I'm going to multiply this by a sum. You see, I'm taking, this is the probability of the viral attack. This is the probability that I have this spacer configuration. And now I'm going to write down the probability that you fail to defend. So first of all, I'm going to write down the probability that you get a particular configuration of Caste complexes given this spacer configuration, right? You give me some sequence of spacers in your CRISPR cassette. They get expressed sort of stochastically, which ones you choose to sort of stick to a Caste. And so there's some distribution that we will derive, for at least in a special case for this thing. I mean, we could derive it generally, but we'll derive it in a special case. And then I'm going to write down 1 minus alpha of V given D. And this is going to be, I'll define all these things now. So first of all, alpha of V and D, let me make this a comma, is equal to the probability of detecting V detecting all viruses in V given the configuration of the Caste complexes, right? You have these machines, these little memory machines, which are Castes armed with a spacer, and these things are going around and checking to see whether there's a virus. And to clear this particular infection, which has 3 viruses in it, I need to be able to get all 3 of them, right? So this is telling you what the probability is, and we'll try to do the statistics of this kind of thing in a minute or tomorrow. Q of D given S, already said, is the probability of producing Caste config D from spacer config S given NP Caste molecules. Right? And there's some distribution there, and we should be able to calculate that. Then there is the PS of S given L. And this involves this probability. This is the probability of having a cassette of configuration S of length L. Now this is tricky because this is sort of statistics. I can play with it. This is tricky because you would think that this may depend on the sort of ecological history of the species, who you've encountered, how you've encountered them, given our previous dynamical discussion, whether your acquisition rate was tuned really high so it was all even, or whether somehow the efficacy of some spacers was much bigger so it was skewed. So there's some issues here. And this is where we're going to take an axe to that problem and simplify hugely. I'm almost done. And then finally, I'm going to write down PV of V. And this is a sad, sad story because this is the probability of encountering phage config V. And to a first approximation, we have absolutely no idea what this is because no one's really studied that, right? So one of the great missing things in a huge amount of the study of biology, of animal behavior, of problems in neuroscience, sensation, perception, many, many things in cell biology and stuff like that, is that we actually don't know what happens out there, right? Almost all the experiments are done in these very special lab conditions. So this thing, which would be great to know, what's the probability of picking up, encountering these different viruses? We don't know. We don't even know that really for the flu and for all the random viruses that people have, and we really care about that. So, right? So this hasn't been done, and this is would be a great goal for any experimental, for any kind of species, figure out what is the distribution of viruses that targets it. But for the moment, we don't know. But as a theorist, I'm free to make up a model that has unknown things and make guesses and assumptions. And we're going to do the obvious thing, which is going to make everything uniform for lack of a better choice. And if you tell me that, no, you should make it skewed, I'll make it skewed. Right? We can study what happens in all these different circumstances. But we're going to study in the blackboard the uniform case. So that's our setup. Our goal will be, after making some assumptions, especially for this one and this one, to make our lives a bit simpler, we will calculate this probability, right? As in terms of some expressions. And then we'll take a derivative with respect to the length of the CRISPR cassette, right? And maximize. And that's going to tell us some interesting things. So that's going to be the beginning of the class next time. And then I'll do another thing. Okay, we'll stop there.